Now that we've gone through the structure of nucleic acids and how we can use that information in the cell, getting from DNA through RNA to proteins, Chapter 12 is a look at how we can apply what we know about microbial DNA to genetics and to recombinant DNA and other biotechnologies. The image on the left is a thermal cycler. That is a machine used to do the polymerase chain reaction. And that reaction allows us to amplify or make a whole lot of copies of DNA. If we were to start with a few molecules of DNA in this test tube in this chamber, we will go through the process of what the machine actually does. But by the end of all the cycles that the thermal cycler would do, we can make billions of copies of a single DNA molecule. We want to do that so that we can detect if even very low levels of a pathogen are present. The example here is Chlamydia trachomatis. It causes chlamydia. It is the most common sexually transmitted disease in the U.S. The reasons for that are large, mainly because of how it presents in differently bodied people. But the test for determining if a person has chlamydia takes advantage of this polymerase chain reaction. A sample can be taken from the patient. We don't have to culture it on agar and wait 24 hours. The DNA can be extracted from that sample and then amplified or copied using PCR. And it's a very specific test, so that if a positive result comes back, it means nucleic acid from the pathogen was found and the person has contracted that illness and can be treated accordingly. This is a very quick picture of recombinant DNA technology. It's called recombinant because we take DNA from two different sources and recombine them into one molecule and then insert that into a cell so that the cell can transcribe that DNA to RNA and then translate the RNA into the protein product. The example shown here is for human insulin. All the people who require insulin to treat their diabetes use what is called rDNA insulin, recombinant DNA insulin. And that was created in exactly this manner. We were able to isolate DNA from a human cell through various techniques. We have figured out the precise gene sequence, which piece of DNA in all of our 46 chromosomes encodes the information to produce insulin. Plasmids are circular pieces of DNA found in bacteria, and we can nick or cut this plasmid, creating ends that are the same as the insulin gene. Specific enzymes are used to do that. And then we can seal this human DNA into the plasmid because, again, DNA is a chemical. The origin, the species of that chemical doesn't really matter. And then, once we have this recombinant DNA, there are various techniques we can use to introduce that recombinant DNA into a bacterium. And when this bacterium grows and divides, it will copy its chromosome, but it will also copy the plasmid so that all the offspring contain this information. This plasmid will be transcribed to RNA and translated to make any proteins that are on the plasmid, but also the insulin. The bacteria are grown in giant vats and large volumes of the insulin are extracted, but it is 100% actual, true human insulin being produced by bacteria. This slide is showing the restriction endonuclease enzyme. It's acting on nucleic acid, and the ENDO prefix says it's acting somewhere inside in the middle of the DNA molecule. It is also called a restriction enzyme. In this case, to restrict means to cut, not to prevent something from happening. And so the restriction endonuclease recognizes a particular sequence pattern within the DNA. And the sites for that recognition are palindromes, meaning the sequence on each strand reads the same from 5' to 3'. So we have our 5' end, and we have GGATCC, and on the 5' end, GGATCC. The strands still have the correct base pairing, but if you read them 5' to 3', they have the same sequence. That is why it is a palindrome. This particular enzyme creates what's called sticky end cutting because it nicks in between this GC pair, and so we have one base pair remaining, and we have 4 that are sticky. They are unpaired. And if we cut DNA and leave it in a test tube, it will hydrogen bond back to itself. But if we cut 2 different sources and we flood a whole bunch of the DNA of interest, we can get our, say, insulin gene to base pair with the sticky ends and create that recombinant DNA. There is also blunt end cutting. We still have that palindrome recognition. We have from 5' to 3' GGCC, and from 5' to 3' on the other strand, GGCC, and we cut in between, and we don't have an overhang. That is why it is called a blunt end. The names of the restriction enzymes come from the bacteria that produce them. Many prokaryotic cells, bacterial cells, produce these restriction endonucleases to degrade the DNA from other organisms, and that is a defense mechanism. Put myself somewhere that is not going to obscure what you need to see. There we go. This big circle is a plasmid, so a circular piece of DNA. It has been very stringently engineered to have features that allow researchers to use it for recombinant DNA technology. This happens to be called PUC19. The P is for plasmid. The UC is for University of California. And 19 because it was the 19th one they came up with. It's fizzy water. So this plasmid has several features. This little rainbow extension out here is called the polylinker site. It's also called the multiple cloning site. And that is because it provides us with several options for a restriction enzyme, a restriction endonuclease, that will cut this plasmid only once. We want to cut it only once because we want that circle just to open up to accept the piece of DNA we're interested in cloning. If we cut it a bunch of times, then it's just a bunch of linear pieces of DNA that are not going to likely reassemble into this nice circle. So this has, what? 1, 2, 3, 4, 5, 6, 7 different options. And we have several different options because depending on the DNA of interest, we may not have all of these options. We may only have one option that will be found with that sequence such that we can cut the DNA of interest and insert it into this plasmid. It has AMP, which stands for ampicillin. So it's the gene for ampicillin resistance. If we take even this plasmid with no other DNA in it and insert it into a bacterium, it will give that bacterium the ability to survive growing in the presence of ampicillin. We do that because that is how we know we are successful in getting this plasmid to enter bacterial cells. The last feature on here is this LAC-Z alpha. That is one of those genes from the lac operon, those genes that are used for digesting lactose. It is called a reporter gene because when we get this piece of DNA into the bacteria, this LAC-Z gene, if we do nothing to the plasmid remains a nice whole circle. The LAC-Z gene is perfect and operational. When we put that in the bacteria and we give them lactose to eat, they're super happy about it. And we put a color indicator that shows that the lactose has been digested and the colonies will turn blue. However, if we cut this plasmid, we also cut in the middle of that LAC-Z gene, and then we stuff in the DNA of interest, and now the LAC-Z gene is broken. And if a plasmid containing a broken or an interrupted LAC-Z gene is put into the bacteria, they still survive because they have the ampicillin resistance gene, but they won't be able to eat the lactose and they won't change colors. So we can use the color of the bacterial colonies on agar to determine were we successful in getting the plasmid into the bacteria, and did that plasmid contain our piece of DNA disrupting the LAC-Z gene. Showing the different steps, we start out with some DNA of interest, and we might not even know the exact gene, but we're looking to find it. So we can isolate DNA from an organism. We also can have the plasmid. You literally can buy a test tube full of liquefied plasmid DNA. We know what the enzymes are that can cut within that LAC-Z gene at the multi-cloning site. So we choose one of those enzymes and we cut the plasmid with it, so the plasmid opens up, cut only once. We use that exact same enzyme to chop up the DNA from our organism of interest. And that will give us a known plasmid with, we know what the sticky ends are. And we know that whatever pieces we get from this foreign DNA, we may not know anything at all about the intervening sequences, but we know the sequence of their sticky ends because those sticky ends were created by cutting with the exact same enzyme. Anytime you use one specific restriction enzyme, you will create the exact same sticky ends on any DNA that you cut with it. And now, because we have the similar sticky ends, if we left these in a test tube and we left these in a separate test tube, they will hydrogen bond and seal those sticky ends. We can't really prevent that from happening. But what we do is we have a low concentration of our opened up plasmid in a test tube, and then we add to that same test tube a whole bunch of our DNA of interest. And so it's way more likely that we're going to get the plasmid stuck to our DNA of interest than the plasmid sticking to itself. We will probably get some of these pieces sticking to themselves, but if they're not in the plasmid, they're not going to get taken up by the bacteria. So we will get a mixture of various portions of this original DNA that's been chopped up with the enzyme inserted into our plasmids. And then we introduce that whole mixture. In our test tube, we have a mixture of all of these different combinations. We can use techniques to introduce that DNA into bacteria. Here it says through transformation. Some cells will do it naturally. Other cells, we can either chemically treat them to weaken their cell walls and force them to take up the DNA. We can also use electricity to blast the cell wall, and then it will take up the DNA. Either way, that is transformation, because we're taking up the DNA in the solution into the cell. And sometimes we'll get bacteria that just don't take up any plasmid, or it takes up these bits of DNA, but it'll be grown on media with ampicillin, and if it doesn't have that plasmid, these bacteria don't survive. Some bacteria will take up the plasmid that just stuck back to itself, and it will survive because the plasmid has the ampicillin resistance gene. The other option is we take up the plasmid that does have one of the pieces of our DNA of interest. That plasmid is good to go. It has the inserted DNA, and so it will also grow in the presence of ampicillin. But the other feature is that LAC-Z gene. The plasmid as it exists, if we don't get anything interrupting the LAC-Z gene, then that plasmid will have ampicillin resistance. The bacteria with that plasmid will grow. They will chow down on the lactose and they turn blue. But if they have the foreign DNA interrupting the LAC-Z gene, they will eat other food in the media, but they can't eat the lactose. And so they don't have the enzyme for that. They don't change the colors, and we end up with white colonies. So on our agar surface, because of the color indicator we add to detect the products of lactose digestion, the ones that have original plasmid, completely intact LAC-Z gene, turn blue, and the ones that have their LAC-Z interrupted by the inserted DNA are white. And so we can literally use our loop to touch just the white colony and streak it on another plate, make sure it stays white, and then we can take from that and grow it in broth and get large amounts of bacteria to continue studying. Feature called genomic libraries means that we extract DNA from some organism and cut it into fragments, just like we did on the previous slide, and inserting it into plasmids and transforming that into the bacteria, and then we will have lots of different types of DNA. Because if we don't know the gene we're looking for, all we know is that we have the correct ends, the overhanging sticky ends, because we cut the plasmid and the genomic DNA with the same enzyme. And then we have to do more genetic studies to figure out which piece of DNA got put into each of these organisms. We talked about transformation when delivering DNA that has been cloned into a plasmid. We can also use transduction. Transduction is using a virus to deliver that recombinant DNA. And in this case, we have some sort of cellular genome, so DNA that came from a cell, and a phage genome, DNA that came from a bacteriophage, a virus. Still have to cut them with the same restriction enzyme, so you end up with the similar ends, the overhangs. But now we're not building a circular plasmid. We're just sticking bits of DNA together. And we can put those in the presence of phage particles, and we will get assembly of both types of DNA into the phage, the bacteriophage. They can infect E. coli cells, and then we can, instead of looking at colonies, when a virus infects a cell, we are going to cause them stress, and it will cause the lytic cycle to happen. So if we plate all of the bacteria that were in a test tube and got infected with the virus and pour that onto an agar surface, it will create a lawn, a solid mass of bacteria. But as the bacteriophage grow and take over the cells to replicate, it kills the bacteria and creates an empty space called a plaque. And each plaque, each empty space, is containing the phages with a unique fragment from the original genome. This looks an awful lot like the previous slide we saw with this little worm, but this is looking at complementary DNA. The reason we need it is eukaryotes have those coding and non-coding regions. The coding regions being called the introns, and the coding regions being called the exons, and the non-coding regions being the introns, because exons get to exit the nucleus and introns stay in the nucleus. So if we extract DNA from this organism, it will have introns and exons in it. And if we tried to put that DNA into a plasmid, it would work. But then when we put that plasmid into a prokaryotic cell, the prokaryote doesn't have the enzymes to cut out the introns. And all of the introns and exons would get transcribed, but then that RNA is not useful for translation. It would have spacers in it that don't code for a product. So instead of directly cloning DNA into a plasmid to put into bacteria, if we start with a eukaryotic organism, like for human insulin, we actually extract RNA, because the RNA will have its introns removed. Then we use an enzyme called reverse transcriptase. Reverse transcriptase is an enzyme that we isolate from viruses, and it has the ability to look at RNA and read that RNA to make a DNA copy. That is the reverse of transcription. So the enzyme reverse transcriptase reads the RNA that has already had the introns removed. It only contains coding DNA, or coding RNA, and it builds cDNA, which is complementary DNA. It is complementary to the messenger RNA. And that complementary DNA can now be cut with a restriction enzyme. The plasmid cut with the same restriction enzyme. We put that complementary DNA into plasmids and transform it and can carry out our experiment. So back to that first slide showing human insulin, that had to be done in this way. we had to have the messenger RNA from humans, that messenger RNA that encoded insulin, use reverse transcription to build the DNA for insulin, and clone that into the bacteria. So electroporation is one of the ways we can force transformation to happen. Remember that transformation is the uptake of DNA from the solution surrounding the cell. We have a bacterium, it has a plasma membrane, and it has a cell wall. Usually the cell wall is treated chemically to either remove it or break it down, and then electric pulse forces holes in that plasma membrane that are big enough to allow the DNA to enter. And then we place the cells into really nutrient-rich media so that the cell membrane can repair itself. This does work. It also kills about 90% of the cells. Fortunately, when this is done, there's more than 1 billion cells present, so you kill 90% of them, you still have 1 billion to hundreds of millions of cells left over. This is a different way of injecting DNA into eukaryotic cells, where we actually have a tiny, skinny needle where DNA is picked up with this microinjection needle and actually punctured directly through the cell membrane and into the cell to deliver that DNA. Now, Dolly the sheep you may have heard of, first mammal to be successfully cloned. This microinjection technique was used to do that. It wasn't through recombinant DNA. The whole nucleus of a mammary cell was removed, and that whole nucleus was microinjected into an egg cell that had had its nucleus removed. So now the egg cell that should have a nucleus with half the number of chromosomes, that nucleus removed, and a nucleus that had pairs of chromosomes from a non-sex cell was put into the egg. That tricked the egg into thinking, it must have been fertilized, I've got pairs of chromosomes, and it started to divide. That created an embryo. The embryo was implanted into another sheep. That sheep incubated that embryo and gave birth to Dolly. For plants, a little tougher because they have that cellulose cell wall, and we can't really remove that cellulose cell wall and have it regenerated. So looks pretty violent. We've got heavy metal particles that are shot using a gene gun. Put the plant's cells in solution and zap it with heavy particles that have the recombinant DNA coated on them. In agriculture, there is a lot of recombinant DNA technology. This is where the term GMO comes from. GMO stands for genetically modified organism. And there are lots of ways to do genetic modification. We kind of do it with selective breeding of plants and animals to get the features we want, but we can also do it through recombinant DNA technology. So we could create transgenic plants because we've taken the gene from one organism and moved it across into the genes of another. So Agrobacterium tumefaciens is an organism that can cause tumors in plants, but we could take the plasmid from that organism and use it as a vector, just like the plasmids to introduce DNA into bacteria can be called vectors. It's the delivery system. So we can clone the DNA of interest, chop it up with a restriction enzyme. We cut the region in the TI plasmid with the same enzyme, We insert the gene of interest into that plasmid and transform it back to the bacterium that generated this plasmid, and we can infect the plant cell with that DNA. This is not necessarily how we get various crops that are genetically modified, but it is one way to deliver genes of interest into a plant. DNA probes, put myself at the top. Am I making you dizzy? There we go. Is a way to look for DNA of interest. In this case, we're looking for a suspected pathogen. If a person has pathogens that can enter body fluids such as the blood or saliva or sexual secretions, then that fluid can be isolated from the patient. And instead of trying to grow the bacteria or the virus, we can isolate the DNA. And there are definitely very sensitive chemical techniques for isolating DNA. And then to get to finding out, does this DNA that we got from a patient sample contain any DNA from the pathogen we suspect causing this patient disease? This would be the entire DNA. Somewhere in here would be, if the pathogen is present, a gene that we know must be present in that pathogen. We can either heat it or chemically treat it to make the original DNA single-stranded. And then we have probes. These are short pieces of DNA that contain the genetic sequence of the gene that we know must be present in the pathogen. And the little stars called molecular beacons, super fancy way of saying we stick a chemical on the end that will fluoresce. It'll glow in the dark a particular color. And these probes are also single-stranded. So if we mix our isolated patient DNA that is single-stranded with these labeled probes that are single-stranded, these two might stick back to each other. These two might just stay in solution. But we will get some where the probe hybridizes or sticks to our gene of interest. And then we can find that with a color reaction, usually fluorescence. Sometimes we can use radioactivity, so we can detect if the sample contains a radioactive tag, whatever the tag is we put on our probe. So this means we don't have to cultivate the microbe. Some bacteria are very difficult to grow. They take days to show up on agar. Some can't be cultivated on agar at all. Viral pathogens absolutely cannot be cultivated on agar. So this is a molecular way to diagnose an illness. If we're looking for particular DNAs, to find out if we got the right piece, or if we have a sample, we want to separate the pieces of DNA by size, we can do something called agarose gel electrophoresis. Electrophoresis means we're going to be passing an electrical current through the substrate. The substrate is agarose gel. OSE tells us it's a carbohydrate, and it is related to the agar that we use in lab, but it is purified more, and it is, when solidified, completely transparent. So we prepare that agarose, and when it's molten, we can pour it into a tray that has a comb. Then when it cools and solidifies, we could take the comb out, and that leaves little divots in the agarose. So that creates a hole or a well for us to place our samples. We could put our DNA samples into these divots in the agarose, and we put them with a dye so that we can see where we put our samples. And the dye also is viscous, and so it makes the sample sink and stay inside that well so it doesn't float out. We take our agarose gel and put it into a tray with a fluid that contains electrolytes so that when we pass electricity through it, we get the DNA to be drawn through that agarose, through that gel substance. DNA has a negative charge just innately because of the phosphate groups. When a phosphate group is placed in an aqueous solution, it tends to give away hydrogens and it becomes negatively charged. So all of those phosphates are negatively charged. The DNA molecule as a whole is negatively charged. So we add an electricity source with the positive electrode at the bottom of the gel so that the particles are attracted to that positive end and they are repelled by the negative end. The DNA separates because of the charge, but it also, because it's moving through this chunky gelatinous substance called agarose, it separates by size. And small particles move faster and they end up at the bottom of the gel. And large particles have a hard time moving through that gooey substance and they stay toward the top of the gel. And what's shown here is the DNA ladder. This would be a mixture of pieces of DNA of known sizes. What we see here, it says 250 BP, 500 BP, up to 2000 BP. BP stands for base pairs. How many nucleotides are present? When we might have dyes in here, it'd be hard to see individual banding. We usually place this whole gel into a fluorescent dye called ethidium bromide. When we put that on ultraviolet light, it glows in the dark. And then we can see here is one ladder. We put another one on the other side for easy comparison, and we could see different size bands appearing in the gel. It looks like no two samples appear to be identical. They have different sized bands in them. We can use this for a technique called restriction, fragment, length, polymorphism. So a restriction fragment, we're going to cut the DNA with a restriction enzyme to create fragments. Length polymorphism means between two samples, the lengths of the DNA created by this very same restriction enzyme will be different. Polymorphic, different shapes, right? This can be done when looking for different strains of bacteria. How much DNA do they share? In this case, a normal versus mutated DNA. It would also be the same technique used when we gather DNA evidence at a crime scene. So we'd look at DNA evidence, or DNA from the victim, DNA from potential suspects, DNA from random people, cut each of those samples with the same restriction enzyme, and we should get different fragment length polymorphisms because none of us are the same. So between this normal and the mutated DNA, we can see that there are many bands that are the same, but there are some that are different. The exposure of this DNA to, looks like radiation, did cause mutation. So not only can we separate the DNA because of its charge and by its size because it's moving through that gooey substance, we can also, in smeared samples on a gel where we really can't see much individual bases, we can use a probe to see if any sequences that we know of are present in the samples we selected. This is called the Southern blot technique because Dr. Southern figured it out. If you digest chromosomal DNA with a restriction enzyme, you do get this big smear. And no matter whose DNA is in what lane, it all looks like a really big smear. But if we wanted to look for the presence of particular DNA fragments, we could transfer the DNA that's in this gel onto a nylon membrane. And not a membrane like a cell membrane, like a filter paper. And we'd place a buffer in the bottom, we put filter paper on there, we put the gel with the DNA facing up, we put the membrane on top of the gel so that as the liquid is wicked by capillary action through all these layers, the DNA exits the gel and sticks to the membrane. And the membrane is not porous to the DNA. It will stick to that membrane. The paper towels are put on top to continue the capillary action in drawing the fluid up. Once we have the DNA stuck to the membrane, if we were to look at it, it would just be the white membrane. There'd be nothing particular on it. But we'd have to put that in a solution with the probe that is complementary to the sequence of DNA we want to see if it's present in this smear, with some sort of marker, be it a fluorescent marker, a color changing marker, or a radioactivity marker. And that would show us that, well, if our DNA of interest is definitely present in 4 of these samples. It's not present in the latter, because that is generic DNA, and we wouldn't want it to have any sequences in common with the probe that we're using. And it is present in the 1st, 2nd, and 3rd samples on the gel, but not in the 4th, and it is in the 5th. This is looking at isolating proteins. The agarose gel electrophoresis isolated DNA by fragments, and SDS is, you won't have to spell this, but it's sodium dodecyl sulfate. It's a detergent. That detergent denatures the proteins, and it also coats them with a negative charge. So to denature DNA means we break those hydrogen bonds and make it single-stranded. To denature protein means we unfold that 3D structure and much of the secondary structure. The gel in this case is upright, and it is made of polyacrylamide. That's what the page stands for, polyacrylamide gel electrophoresis. The gel is poured between 2 glass plates. There's a comb in it. When we take the comb out, that creates the wells. We put the protein sample in each well with a dye so that we can see it. Once again, we have a power source, and this is put in a buffer. We've forced the proteins to be negatively charged, so they will migrate toward the positive pole. And then we can stain it with particular stains and look at the banding patterns, just like we would for DNA, but this is protein. This shows up because it is the classic bullseye rash that shows up in many patients who have been bitten by the tick that transmits Lyme disease. But as it says in the caption, up to 30% of infected individuals never get that rash. And so their disease can progress quite far along before they get diagnosed. If someone suspects they could have been exposed to a Lyme disease tick, we can isolate blood from these patients and do a PCR test to look for the presence of the DNA. The polymerase chain reaction, the PCR test, is that test that makes lots and lots of copies of a specific region of DNA. You isolate DNA from patient fluids, from a bacterial culture, from plant culture, whatever it is that you're looking for. You can do a chemical isolation that will give you the DNA, and then the thermocycler takes it through the steps of DNA replication in a test tube. The first step is denaturation, so that we get single-stranded DNA. In a cell, we don't do denaturation. We have the helicase enzyme that breaks the hydrogen bonds a little bit at a time. But this is not a living cell. This is just the chemicals. And so we can crank the heat up to 95, and that makes all the DNA single-stranded. Then we have primers that are short stretches of DNA that we know the sequence of, and we know that it will give us the ends of this gene that we're looking for. So if we know nothing about the DNA sequence of the starting material, we can't do PCR. We have to know something about the DNA sequence. We can use DNA that's been put in a plasmid because we know the entire sequence of the plasmid, and we can use primers that start in the plasmid, and then the replication will go through where the insert is in the plasmid. But we have a primer to each side, each strand of the DNA, and then we change the temperature to allow the DNA polymerase to extend and go from the end of the primer, which will have a 3' OH, and make the new DNA strand, and it will do the same thing on this primer, bind to the 3' OH, and generate that strand. And at the end of the first cycle, you will have 2 copies of DNA, one with each original strand, and each of them has 1 new strand. In cycle 2, we take each of these molecules, so the one with the new red strand on top, the one with the new green strand is here. We heat them up, so we pull the strands apart. We add the primers, we extend, and now our original strands are still present, but we've got 2 molecules that are both new strands, and we have 2 molecules that have an old strand and a new strand. Old strand and new strand. We repeat this over and over time, between 20 to 32 cycles, and every 1 cycle doubles the number of DNA molecules present. We're never going to have a single DNA molecule present at the start, but if we did, after 1 cycle we would have 2 copies, and after 2 cycles we have 4 copies, and after 3 cycles we have 8 copies, and after 4 cycles we have, 16 copies. We double the number of copies every single time. So if we do 20 cycles, that would be 2 to the 20th power number of DNA molecules. So we can create an enormous amount of DNA. And to do 20 to 32 cycles takes around 3 hours. It's not that long. This is showing how one of the coronavirus tests is done. There are 2 different tests. The nasal swab test is this test. It uses PCR. The other test is called the antibody test. And so they would extract from your blood and look to see if you have antibodies for the virus. This test is very sensitive because you don't need a whole lot of virus. The coronavirus is a positive sense single-stranded RNA virus. That means our cells would recognize it like messenger RNA. So our cells would be able to translate that viral genome and make proteins, but PCR requires DNA. So from the nasal sample, and that is how far back some of them go. I've had 2 tests. The first one, they went all the way back. The second one was a lesser swab. Either way, that swab is put into a solution to get any of the cells and any viral particles into that solution, and then the viral genome is isolated, and then reverse transcriptase has to be used to get a DNA copy of that genome, and then PCR is used on that viral complementary DNA, and there are tags put on the primers so that we can see that there is a positive result. These dideoxynucleotides are shown here because they are used for getting the DNA sequence. If we start with DNA of unknown nucleotide sequence, we can figure that out. A normal deoxyribonucleotide has the sugar, the deoxyribose, and its 3 phosphates, and it has its base attached. A dideoxy has another oxygen removed. So it's deoxyribose because it doesn't have an oxygen here. It's dideoxyribose because it has no oxygen here. This is useful in DNA sequencing because when we do make a polymer of DNA, we rely on this 3' OH being available for the DNA polymerase to add the next nucleotide. If we don't have an OH, we just have an H, DNA polymerase will no longer extend this. These dideoxynucleotides are used in a form of DNA sequencing called chain termination because we end the chain of nucleotides once a dideoxynucleotide is introduced. This DDCTP, DDATP, etc. stands for dideoxy cytosine triphosphate, dideoxyadenine triphosphate, dideoxyguanosine triphosphate, and dideoxythymine triphosphate. So CTP, ATP, GTP, and TTP are the normal nucleotides, but dideoxy means we have those nucleotides that have both of their oxygens removed. The way this is set up is you have 4 test tubes. In this test tube, you would have 4 normal nucleotides, but your 3 normal nucleotides. Your 4th one would be a dideoxythymine, and it's got a fluorescent label that's red on it. This one would have normal A, C, and T, but would have a dideoxy G with a different color on it. This tube would have normal C, G, and T, but its A's would be dideoxy. Normals G, A, and T, but its C's are dideoxy. You mix this with single-stranded DNA that you don't know the sequence of. You mix it with the nucleotides to build a new strand and the polymerase to allow that to happen. And as the DNA is built, anytime the dideoxynucleotide gets introduced, the chain terminates. And as this chain is extending, a machine reads which nucleotide did we end at. And so this would say this nucleotide is a G, this one is an A, this one is a T, this one is an A. So by the color of the peaks, we can read what nucleotide was present when we ended. So as we continue, it's getting longer and longer. We know which nucleotide was at the end, and we can read that sequence off. So there's that Sanger sequencing. We have the different colored nucleotides that will terminate, and as the fluorescent laser detects which color is coming through the gel, it prints that out on the computer so we can read the sequence of the DNA as it's being synthesized. This is looking at something called PCR ribotype. It's called a ribotype not because we're using RNA in the PCR, but because we are using DNA primers to the genes for the 16S size and the 23S sized ribosomal RNA. So the DNA that encodes the information that when it's transcribed will be ribosomal RNA, that is the DNA that we're looking at. This is looking at Clostridium difficile strains. Clostridium difficile is the microbe that causes C. diff, or Clostridium difficile colitis. Can be extremely damaging and even deadly. There are many different types, ribotypes involved. So they're all the same genus and species, but there's variations within them. And so we can look at these known ribotypes, and then we have a patient's ribotype, not of the patient's ribosomes, but the DNA from Clostridium difficile contains these particular genes for ribosomal RNA. That means he has the same as reference ribotype 027, and we know how to handle these particular strains, what there is about them, so knowing the patient's specific ribotype can help with treatment. Another way to identify them can be with ribotyping. Already had that. And when we look at the gel, this is what an actual ribotyping gel can look at. We look at the DNA ladders so that we know what the sizes are and we can compare the different strains. So the eukaryotic cells we mentioned before, they do have both coding and non-coding regions in their DNA. Their RNA has removed those non-coding regions. But there are ways to control protein synthesis other than making the right messenger RNA. This messenger RNA contains the codons that should be met with the ribosomes to get translated. However, cells can regulate, even if this messenger RNA is present, they can create something called antisense RNA. That means it has the complement to the messenger RNA, and that blocks the ribosomes from being able to translate that message. So this is not genetic engineering. This is something that naturally occurs in our cells that we can control whether or not this messenger RNA actually gets translated. Gene therapy is another technique where we can use our knowledge of microbial genetics. We have the DNA that has the good or healthy gene in it that we can clone into viral DNA and insert that into a viral vector that can enter into the cell, and then the recombinant viral genome will enter the nucleus of the host cell and try to incorporate and get copies of it made to try to treat patients that have genetic illnesses. So far we have limited success because the virus has to infect, the DNA has to get in, the DNA has to incorporate into the host genome. That was a whirlwind tour of microbial genetic applications. I'm going to find some more videos to post, both for that DNA replication from last week and for these recombinant DNA techniques that we've seen in Chapter 12.